So Bo and I are about to plant our cheatgrass experiment where we're going to look at the potential role of a cheatgrass pathogen that we found this summer to treat cheatgrass, so get rid of cheatgrass. So what we have here are 16 plots in 4 blocks, and we have cheatgrass seeds that we collected on the ranch that we've surface sterilized to get rid of the pathogen. And the one treatment is just a control. It's an untreated seed. And we have the positive control here that we've treated with this Ustilago bullata. It's basically a bunch of spores that we've coated the seeds with and hoping to get an effect of that. But obviously, to combat cheatgrass, we can't dig up all the seeds and coat them with the spore solution. So what we're trying to do is figure out a way to apply this on germinating seedlings. So 2 treatments here, we're going to either apply a spore solution directly on the seedlings or grow them in culture. So you actually have the vegetative growth of the fungus, and Bo knows a lot more about this than I do. And apply that, because that could be a management tool where we can actually apply it on large scale. So we have 4 replications, and we're going to see if it works in the spring.
All right, we're on the north end of the MPG Ranch property here. This is continuing with the Ustilago bullata biocontrol project of cheatgrass. And this is a study plot here, a naturally growing stand of cheatgrass. We haven't planted any seeds here. But in September, Ilva inoculated this with Ustilago sporidia. And we're going to inoculate again with the same sporidia here in November to see if there's a seasonal difference on infection. We're also just generally trying to learn if you can infect grass that's growing on the soil with Ustilago. So what are sporidia? We have these liquid cultures here, and this is malt extract broth, which is just a carbohydrate-rich solution that fungi can grow in really well. And we grew some Ustilago spores on agar plates and then took the resulting kind of slimy mycelium from that and put it in this broth and then let it sit for 5 days or so. And it would have been transparent before, but you can see it's all milky because of all the fungal culture that's in there. So this is part of the life cycle of Ustilago. They have spores that germinate on the cheatgrass seeds. And then when those spores germinate and start growing, they grow this kind of hyphal mycelium that's in here now. So we want to know if we can infect cheatgrass with this, because it's a lot easier to grow this than it is to collect teleospores. So we've grown this. This is a really thick culture. We don't need to spray it on the cheatgrass this thickly. So what we're doing is we're going to put it in here at a 10% dilution into just plain water. And then we'll mix it up and spray it onto the cheatgrass. So that's 250 mils. This is another 250, so we'll have 500 milliliters in here with 4.5 liters of water, and we'll have our 10% dilution. Ready to go. And we want to spray the same amount of this on each of these plots. These are 2 meter by 2 meter plots. We want 400 milliliters of this solution to go on each plot. So we've timed that out. If I spray for 80 seconds, we're pretty close. So let's get to it. So we've just finished spraying the Ustilago inoculum on these plots here. And one reason we chose this plot on the north end is that we didn't observe any Ustilago infection in these seeds. So this is sort of a control. We want to see if, we're assuming that the fungi that we introduce here are novel. In other words, when we count infected plants, we're not counting ones that were already infected the year before, or that there were already spores here. So it's a control plot for us. And we'll wait a year or so until the new crop of cheatgrass produces seeds. Then we'll be able to count and see what kind of infection rate we got. In some of our videos in the field with this Ustilago bullata project, we talked about how the infection in the field of cheatgrass by Ustilago is patchy. Some cheatgrass stands are infected and some aren't. Well, one reason could be that there's genetic differences in this fungi. Some of them may have the genetic ability to infect cheatgrass and others may not. So one thing we've done to try and figure this out is take cheatgrass seed samples that are infected with Ustilago from 10 different sites on the property. And then we want to obtain cultures and DNA sequences from these different plots and then be able to look at them for genetic variation. This part of the project, our high school intern from Big Sky, Stacia Hill, has been working on. And she's not available to do this video, but I wanted to make sure and acknowledge her work on this. She's been culturing this Ustilago from these plots for our genetic evaluation. So how does this look, the work for this look like? What we need to do is obtain pure cultures of each from each of these 10 plots. So what I want to do is get some Ustilago spores out of here and then onto a nutrient media plate like this where it's growing. The way I know it's a pure culture of just that particular genotype of Ustilago. So what we do to make this happen, we get a little fluid on a slide, a little water, and then I need a clean tool to work with. So what I'm going to do here is grab just a single seed, and what I'm doing, you'll notice that for each plot, each bag of cheatgrass seeds that we've collected here from the field, we're ending up with 5 different plates. So what I'm doing is we're collecting seeds from 5 different stocks so that we do have replication. That way we'd be able to say how much difference is there genetically between Ustilago, between plants of cheatgrass, and then how much diversity does that compare to how much there is between all the plots. So it ends up being a lot of plates. So the next step, we've got some water on this slide, some sterile water. I'm using clean tools here. And we're just going to try and scrape a few of these spores, and you won't be able to see this in the video. But we scrape a little bit, and we have some spores land in that water, and they're there. And then the next step, we actually have to get the spores out of that fluid and into a nutrient plate, but we need to make sure we only get those spores. There could be other fungi on this grass, and we don't want contamination. So we open our flow hood here. It starts making a lot of noise. I hope you'll still be able to hear me. This forces sterile air in at a constant pressure so that spores, other contaminants, even like my breath and stuff that's in my hair, can't get into this workspace. So just use a simple dissecting scope. One little piece. And you can see the spores sort of adhere to this little spatula. And then take my media plate, stick the spores in there, and it's done, they'll grow. So all I need to do now is wrap this to make sure it stays moist and that no contaminants enter. We'll label it, which plot it came from, and just number it for which different stock that fungus came from. And after they've grown for a while, what we end up with over time is these cultures here. And, you know, despite your best efforts, you can end up with problems. You know, you can see the difference in growth between this one and this one. So, you know, that first one might be a contaminant, and this one certainly is because it's a different color and it's kind of a fuzzy growth. So then you have to go back and reculture a few of them. But these next ones, I mean, that's exactly what Ustilago should look like in culture. And you know, after about a week or so, you know, you see enough growth to know what's going on your plate. So we should be relatively close here. Most of the culturing's done. We just have to go back and make sure and take care of our contaminants. And then we'll be able to start running some genetic tests.